Today's researchers face the challenge of measuring low levels of specific proteins in biological samples containing tens of thousands of different proteins. The AMMP assay works at a molecular level using sound versus optics to both sensitively and accurately detect and quantitate proteins, regardless of the sample complexity. Here, we'll illustrate the mechanism of detection using a sandwich assay, a sample analyte between two specific antibodies. The AMMP assay begins in the well of a standard microplate where a sample containing an analyte like the protein interleukin-6 is present. Magnetic beads with capture antibody attached are then added. Thousands of antibodies can be attached to each bead. Finally, a paired second antibody tagged with a universal hapten is added. During incubation, a complex of magnetic bead with antibody, the analyte, and a paired antibody hapten is formed. Following incubation, samples are transferred from the microplate by the Vibe workstation to a Vibe cartridge, one column at a time. Like a column in a microplate, the Vibe cartridge consists of 8 sample wells, each with a discrete fluid path. The samples flow into a specialized membrane sensor for direct detection of the protein or analyte. Unlike optical techniques which have issues such as light scatter, fluorescence interference, or quenching and use indirect measurements, BioScale has a simpler approach. We measure the frequency shift of a vibrating membrane caused by the interaction of an analyte on the sensor surface. A magnet is engaged as the analyte magnetic bead complex appears in the flow path, enhancing the attraction and binding of the beads to the sensor surface, thus slowing the vibration of the membrane. The beads containing the analyte complexes bind with the antibodies on the sensor surface, while the rest of the beads are attracted only by the magnetic field. The buffer solution is changed. When the magnet is released, the buffer flow carries away non-antibody captured beads, and the vibrational frequency slowly increases. The signal readout depicted in this trace shows you the direct measurement of the protein concentration in the sample. By progressively increasing the flow rate, the remaining antibody-bound complex elutes, and the sensor's vibration returns to the original levels. The surface of the sensor is regenerated with a series of buffers to prepare for a new sample. Regardless of sample matrix, the AMMP assay's ability to ultra-sensitively measure minute changes of frequency without interferences or signal scatter provides robust, highly precise, and consistent quantitative data, experiment to experiment.